Welcome to ASRS's Journal of Vitreoretinal Diseases Authors Forum. I'm your host, Dr. Timothy Murray, Editor-in-Chief of JVRD. On each episode of the JVRD Authors Forum, I will interview innovative retinal researchers on their studies featured only in JVRD and how these studies will impact our patients' care in our clinics. Tune in to hear directly from investigators about the clinical implications of the newest and highest quality research in the field of retina. Welcome to JVRD's Author Forum podcast. I'm thrilled to be joined by Dr. David Eichenbaum, who is a partner and director of research for Retina Vitreous Associates of Florida in St. Petersburg, Florida. We'll discuss his manuscript, Biosimilar Ranibizumab, Ranieyes, Safety and Efficacy in the Real World, the BRESER Study. Welcome, David.
Happy to be here, Tim. Thanks for inviting me, and it's a privilege to have the paper published in JVRD, and full credit to the lead author, Ashish Sharma, for organizing this worldwide group to look at real-world use of biosimilars.
Well, I think you and I agree that particularly in the biosimilar world, that we do need real world data to look more broadly, potentially at the outcomes and impact, and even sometimes worries about safety. Can you tell me a little bit about how does a biosimilar come to FDA approval that differs from the original molecule?
Yeah, certainly. And your sentiment is kind of, that's the punchline. That's the end of the sentence. The beginning of the sentence is exactly what you're asking me to describe, which is how do biosimilars come to market? It's best visualized in my experience as the inverted pyramid versus the upright pyramid. The inverted pyramid is how we develop traditional drugs. We have lots of data and we narrow them down and we have a traditional drug at the very end. With biosimilars, we have these traditional drugs and there's a very broad base to the pyramid looking at creating a product that is similar to the originator product on a chemical level, a molecular level. There's a lot of basic science and a lot of comparative chemistry that goes into the approval of a biosimilar in the US and worldwide in a similar process. And the FDA looks at that. And then at the top of the upright pyramid, the biosimilar development pyramid, you have a very short in-human clinical study. Usually it's 2 to 3 months. Primary outcome is 6 or 8 weeks with a short safety extension after that in some studies. And then the drug is approved. It's usually a relatively small population, usually compared to the originator, but you don't have a whole lot of clinical data to go on. And this is not just true for retina. This is true for the therapeutic areas where biosimilars really started in the US, which is cancer and immunology. And what you then have is a drug that we need to learn about in the real world.
So it's always been interesting to me because I think when people started hearing about biosimilars, they thought more of what a generic drug is like in the pharmacopoeia that we deal with, where, are you taking Advil, or, and those drugs to me are very straightforward in the manufacturing process. But biosimilars, by their nature, we're really using a complex organic process to build these molecules. So how do we get through that phase of development so quickly once there's an originator molecule?
Yeah, so I think that has to do with the stringent and precise nature, almost the digital nature of monoclonal antibodies that are made into biosimilars versus the small molecule nature of drugs like ibuprofen, Advil, what have you. I think that there is more precision involved in the approval process for the monoclonal antibodies. And once you achieve that similarity, which takes time, I think, to reverse engineer the monoclonal antibody once the patent expires or you're willing to try to go head to head with a drug that's an originator at any point in its life cycle. I think once that drug is reverse engineered and all that work is put in, I think then you have a relatively rapid path to approval as you sort of move up the biosimilar pyramid.
But did you have concerns that we really have looked at this unique drug, even though it's a biosimilar, in such a small proportion of patients? Um, that's kind of the opposite of the way we think we would like to look for safety,
Mhm.
even more so than efficacy, correct?
Yeah, certainly. And there are concerns because of the small population when a drug comes out as a biosimilar. I think those concerns are mitigated some by the path to approval, that there's a lot of um, more preclinical work, I think, that goes into determining the similarity. But in India, for example, there was a biosimilar called Razumab that had problems with inflammation because of components of the excipients with the drug. And then that had to do with the regulatory environment in India. And I think in the US, we have a different regulatory environment where we have a lower risk of having inflammatory excipients related to biosimilars. But regardless, to determine that the drug is safe in a broad population with the biosimilars, we do have to look at real world evidence because we just don't have that population in the FDA's clinical development pathway for biosimilar products.
Yeah, and even for one of our more recent drugs, brolucizumab,
Mhm.
we didn't see the severity of complication that we were
Mhm.
able to experience it rapidly in the real world. And then we were able to go back and look for that in the
Mhm.
clinical trial data. But to me, I always think of these biosimilars under that sort of
Mhm.
mantra of we had a drug at market that we missed a potential blinding complication for.
Yeah, we had developed brolucizumab through the traditional pathway, the Hawk and Harrier paired phase 3 randomized controlled trials, and then there was programs for diabetic macular edema and so on and so forth. We were involved with all those programs, and it took the larger real world population to really detect the infrequent yet serious safety signal related to brolucizumab administration. And that's what real world data is valuable for, whether it's for a drug that had 1,000 plus patients exposed to it in clinical trials like Brolo, or a drug that's had several dozen to a couple hundred patients like a biosimilar exposed for a longer amount of time. The real world population is large, and that comes with confounders, that comes with an absence of um, uniformity. It comes with an absence of randomization, but it comes with volume. And there's something to studying a larger population, a voluminous population, to determine the safety of these products. But because of the biosimilar development pathway and the basic science that goes into it, we in our practice have become earlier adopters of biosimilars since we learned about them and had experience with the first ranibizumab biosimilars and now with the aflibercept biosimilar marketed by Amgen in the US.
One of the things I think that really drives the biosimilar market is potentials in cost differential.
Mhm.
Were you a little bit surprised how highly priced some of the initial biosimilars were when they launched before your expectation as to what you thought they would launch at?
Yeah, so it's a really interesting thing because biosimilars do not offer clinical innovation, right? That's the whole job of the originator drug, to offer clinical innovation, usually more than a decade before the commercialization of the biosimilar. So the entire thesis of a biosimilar is economic. It has to do with saving the system money. It has to do with saving patients money. It has to do with increasing the accessibility of a specific established originator drug to a larger population. And I think that the strategy with the biosimilars is related to the pricing. And I was surprised that the pricing was high. If you look at the ASP of the ranibizumab biosimilars 3.5 years after they've come to market, it's become very low very quickly. I think that's how the biosimilars work with regards to pricing. But one would think that with the goal to improve access vis-a-vis reducing cost, those prices would come in maybe lower to start with.
Back to your paper. So we've talked about these,
Mhm.
really philosophical principles of how we develop drug and how we evaluate, how we potentially market, what we price at. So in your study, the take-home message really for Ranieyes
is that it's safe in a large population. It's not a tremendous population either. This is several hundred eyes in the paper in India where the drug is marketed. But the take home is that it has a risk of adverse events similar to what we see in the originator molecule in ranibizumab.
And do you think that we're going to see this as a strategy going forward, that we have originator molecules that are developed maybe in the US, very expensive development course, they're protected for a small period of time, and then we're going to see for every one of those molecules a biosimilar?
Well, I don't think it'll be for every one of those molecules. I think the biosimilar manufacturers will look at the ones most likely to succeed in their markets. When a drug is developed at a high cost in the US, it often is either commercialized by the development company or the commercializing company internationally, or by a licensing partner. And those international approvals are strategic as well. They want to have approval and protection in various countries to return on their initial investment. And you're right, that period of protection is different country to country. But I think it's a combination of strategy from the originating company and strategy from the biosimilar company to overall increase the availability of these drugs around the world, which is an interesting thing to observe, and not something that I'm an expert in with regards to strategy, but something that is interesting to see as our first generation branded drugs kind of come to the end of their branded life cycle.
No matter what, it's an exciting time for us as retina specialists, and the access that we have for our patients is huge. So David, thank you so much for taking time to talk with us and with our audience and elucidate some of the issues that are going to continue to play in the biosimilar world. I think you did a beautiful job explaining that. And thank you for your paper to JVRD. We'll reference that going forward also. Thanks for tuning in to the JVRD Authors Forum. You can watch and listen to more episodes on the ASRS YouTube channel and on popular podcast directories, including Apple Podcasts and Spotify. Visit www.asrs.org/JVRD Forum on the ASRS website to learn more. See you soon.